Hi everyone, I'm Lisa. I'm one of the fourth year medical students, and today I'll be giving a revision lecture on gastrointestinal physiology. So today I'll be covering swallowing and GI motility, GI secretion, absorption, egestion, and GI defense mechanisms. And just for your own reference, anything that's clinical in here will be in pink. So starting with swallowing and GI motility. So swallowing is also known as deglutition, and that's something that's good to know for Part A's because they might refer to that instead of using the word swallowing. And you can divide swallowing into three phases. So the oral phase is the phase that takes place as soon as you put food in your mouth. Then the pharyngeal phase takes place in the oropharynx, and the esophageal phase takes place in the esophagus. And so starting with the oral phase, when you put food in your mouth, the first thing that you're going to do is start to chew it. And mastication is just another word for chewing. And once you've done that, you then need to trigger the swallow reflex. And the way that happens is that 3 out of the 4 extrinsic muscles of the tongue, so genioglossus, palatoglossus, and hyoglossus, will move the bolus of food onto the posterior third of the tongue. And that stimulates cranial nerve 9, or the glossopharyngeal nerve, to trigger the swallow reflex. And the whole of the oral phase is under voluntary control, which is easy to remember because you can obviously control your own chewing. I think the bit that's difficult to get your head around is that the swallow reflex itself isn't voluntary, but it's the contraction of the extrinsic muscles of the tongue that initiate the swallow reflex. And that contraction and the initiation of the swallow reflex is under voluntary control, even if the reflex isn't. Okay, so once you've triggered the swallow reflex, the next place that the food goes to is the oropharynx. So once food's in the oropharynx, if you think back to your anatomy, right above the oropharynx you have the nasopharynx, and right below the oropharynx you have the esophagus and larynx. And you want the food to go from the oropharynx into the esophagus. What we don't want to happen is for food to go up into the nasopharynx or down into the larynx, where it could end up in the lungs. And so the way that's prevented from happening is that the soft palate has to move up to seal the nasopharynx. And then you have the epiglottis covering the opening of the larynx. And that's done by this muscle called mylohyoid. And so once you've covered up the nasopharynx and the larynx, the food can only really go down into the esophagus. And yeah, so when the styloglossus pulls the tongue up and back, that forces the bolus of food into the pharynx. And then once the food's in the esophagus, you have the esophageal phase, and that's basically just peristalsis. And peristalsis is the classic wave-like motion and contraction of the circular and longitudinal muscles, which propels food in one direction down into the stomach. And if you're ever talking about this in an essay, don't forget to mention the upper esophageal sphincter at the top of the esophagus and the lower esophageal sphincter at the bottom of the esophagus. And again, in an essay, you might want to mention this condition called achalasia. And if you have achalasia, you basically have increased tone in the lower esophageal sphincter. And so when you swallow food, it doesn't open up as much as it should do. And as a result, food can't pass down into the stomach and it starts to back up along the esophagus. And that's really dangerous because if food backs up through your esophagus, it can end up going down your larynx and down your trachea into your lungs, and you can end up with an aspiration pneumonia. So that's a good disease to learn about. So GI motility. In your GCSE and A-level biology, I think we're usually taught that peristalsis happens throughout the GI tract, but actually it's a bit more complicated than that. So peristalsis happens in the esophagus, and in the small intestine, the circular muscles and longitudinal muscles are still contracting, but they don't form this wave pattern of contraction, and instead they contract to cause segmentation, so that's when the circular muscles contract, and pendular movements, which is when the longitudinal muscles contract. And so what's the difference between peristalsis and segmentation and pendular movements? Well, peristalsis, if you think of a toothpaste tube, peristalsis is basically if you were to start at the bottom of the tube and slowly push the toothpaste out towards the top in one direction. So the aim of peristalsis is to move food along in one direction from one place to another. Whereas segmentation would be the equivalent of squeezing the toothpaste tube right in the middle. And so you're forcing toothpaste in two directions. And then pendular movements would be if you took the cap in one hand and the end of the toothpaste tube in the other, and then just moved your hands together and then apart again. So segmentation and pendular movements basically mix the chyme that's come from the stomach with the secretions of the small intestine. So it kind of makes sense why that happens in the small intestine more than peristalsis does. Okay, and then finally you have in the large intestine, peristalsis, segmentation, and pendular movements all occur together, but you have less segmentation and pendular movements compared to the small intestine. Great, and all of these movements are coordinated by the enteric nervous system. And if you're ever talking about this in an essay, a good disease to mention is Hirschsprung disease. And that's where you have an aganglionic segment of colon. So you can imagine that if you don't have any innervation from the enteric nervous system in just one segment of colon, then that part of the colon is not going to be able to move food along. So you end up with constipation. And people with Hirschsprung's disease need surgery ultimately to correct this. Moving on to salivary secretion. So your syllabus wants you to know about major salivary glands and minor salivary glands. So there are three major salivary glands that you need to know about. The first is the parotid gland, and that's the really big salivary gland in the cheek, and that produces serous secretions and amylase. Then you have the submandibular glands, so under the mandible, and those produce both serous and mucus secretions. And finally, you have the sublingual glands, so under the tongue, and those produce mucus secretions. Then you have thousands of tiny minor salivary glands throughout the mouth and on the lips, and those produce mucus secretions mainly just for lubrication rather than digestion. So how is salivary secretion controlled? Well, you need to know the parotid gland is controlled by the 9th cranial nerve via the otic ganglion. And then the sublingual and submandibular glands are controlled by the 7th cranial nerve or the facial nerve via the submandibular ganglion. And the way I like to remember this is that parotid has an O in it, so it's the otic ganglion. And the sublingual and submandibular glands, so they both start with an S, so it's the submandibular ganglion. I think that's a classic MCQ question in Part A's. And if you're ever writing an essay, you might want to structure it a bit differently. So you might want to talk about parasympathetic and sympathetic control. So the parasympathetic nervous system, that's your rest and digest, has the largest effect on stimulating salivary secretion. And those parasympathetic nerves release acetylcholine onto M3 receptors. And M3 receptors are GQ coupled. So that's the PIP2 IP3 pathway. IP3 binds to IP3 receptors on the endoplasmic reticulum, and that leads to the release of calcium into the cytoplasm. The calcium then activates protein kinases, which phosphorylate the cytoskeleton, and that leads to the insertion of chloride channels into the apical membrane. And so you can imagine how once more chloride ions move out of the cell, water will follow, and so that increases watery secretion. On the other hand, the sympathetic nervous system has less of a stimulatory effect on salivary secretion, which makes sense because it's fight or flight, and that's the opposite of rest and digest. So in the sympathetic nervous system, noradrenaline acts on both alpha-1 and beta receptors. Alpha-1's GQ coupled, so it's exactly the same mechanism as before. You get calcium release into the cytoplasm, and the calcium activates protein kinases, phosphorylate the cytoskeleton, you get the insertion of apical chloride channels, and so you have increased secretion of saliva. However, when noradrenaline binds to beta receptors are GS coupled, so that's the adenylyl cyclase converting ATP to cyclic AMP, and that ultimately results in an increase in amylase secretion and an increase in the secretion of viscous saliva. So that was quite a lot of information, but the key things that you should remember from this are that the parasympathetic nervous system increases watery secretion via acetylcholine, and the sympathetic nervous system has a relatively small effect on salivary secretion, but it both increases secretion and produces mostly viscous saliva as opposed to the watery secretion you get with the parasympathetic nervous system. So moving on to gastric secretion. Gastric secretion comes from gastric glands, and these glands are composed of a variety of different cell types. So first you have the parietal cells, which are found at the base and the neck of the glands, and those produce hydrochloric acid and intrinsic factor, which is involved in vitamin B12 absorption. Then you have the chief cells, which are also found at the base and the neck of the glands, and those produce pepsinogen, which is activated by the gastric acid to form pepsin. You also have endocrine cells at the base of the glands, and those produce gastrin and somatostatin, which we'll talk about in a second. You also have superficial epithelial cells, which produce mucus and bicarbonate, and you have mucous neck cells, which produce mucus. And if you don't remember all of these different cell types, the ones to remember are the parietal cells, because those are the ones that are responsible for acid secretion into the stomach. So this is quite a simple diagram that you can reproduce in an exam, and it sums up the whole mechanism of gastric acid secretion in a parietal cell. So this shows one central parietal cell with two parietal cells either side of it, and they're joined by these black circles which I've drawn, and those represent the tight junctions. The left-hand side is the apical side, and the right-hand side is the basolateral side. And you know that because the tight junctions are closer to the apical side. So the tight junctions are dividing the cell surface membrane into just an apical side and then basolateral. So just looking in one parietal cell, first you have water and carbon dioxide being converted to carbonic acid by carbonic anhydrase. And then the carbonic acid then dissociates into hydrogen ions and bicarbonate ions. And the hydrogen ions are pumped out into the lumen via this H plus K plus ATPase. And because you're using a potassium ion and pumping it into the cell, that potassium ion needs to leave somehow. So that's what this potassium channel is for. Then you have the bicarbonate, which is also produced, and that leaves via a basolateral bicarbonate chloride exchanger. And because you're bringing in chloride, that chloride also has to leave. So that's the other channel that's drawn here. So how is gastric acid secretion controlled? Well, we can start with the ECLs, or enterochromaffin-like cells. And these cells produce histamine, which acts on H2 receptors on the parietal cells. And the parietal cells are the cells that we just talked about with the H plus K plus ATPase. And so when the H2 receptor is activated, that leads to the insertion of ATPase, of the H plus K plus ATPase into the apical membrane, and that therefore increases gastric acid secretion. So if we want to control gastric acid secretion, we can either act on the ECLs or on the parietal cells. The ECLs can be controlled by gastrin, which is produced by G cells in the stomach. And gastrin acts on the CCK-B receptor, or the cholecystokinin B receptor on ECLs, and that stimulates the release of histamine. You also have a stimulatory effect from the vagus nerve, and the vagus nerve is part of the parasympathetic nervous system, so it uses acetylcholine. And acetylcholine released from the vagus nerve binds to M1 receptors on the ECLs, and that also stimulates histamine release. And the vagus nerve can also act directly on the parietal cells. If the acetylcholine binds to M3 receptors on the parietal cells, then that will also increase H plus K plus ATPase insertion. And something that you shouldn't forget about is somatostatin. So somatostatin has an inhibitory effect on the G cells, the ECLs, and the parietal cells. So this is a diagram that I would definitely just learn and try and reproduce in an essay, because it just sums everything up really nicely and saves you a lot of time and word count. So if we are writing an essay about this, you can mention Zollinger-Ellison syndrome, which you might have heard of. That's where you have a gastrinoma, usually in the pancreas, and that gastrinoma secretes excess gastrin. So excess gastrin means excess histamine release from the ECLs, and therefore excess H plus K plus ATPase insertion and excess gastric acid secretion. If you have excess gastric acid being secreted, that acid can damage the lining of the stomach, and as a result, you can have peptic ulcers. Another cause of peptic ulcers is H. pylori or Helicobacter pylori, and that's a bacterium that inhibits somatostatin release. So if you have less somatostatin, then you have increased activity of the G cells, ECLs, and parietal cells. So that's the mechanism of action for how H. pylori causes peptic ulcers. And the way you treat H. pylori is by giving a PPI, proton pump inhibitor. So the proton pump is the H plus K plus ATPase. So if you inhibit that, then you're reducing gastric acid secretion, and so you're reducing the amount of damage to the stomach. You also give 2 antibiotics, and that's to target the actual cause of the peptic ulcers. And a final cause of peptic ulcers are NSAIDs. And these are drugs that inhibit the cyclooxygenase enzymes. So you want to inhibit COX-2, but these drugs also inhibit COX-1, and that leads to a reduction in the protective lining of the stomach, the mucus layer. And that's the mechanism of action of NSAIDs causing peptic ulcers. So NSAIDs don't actually increase gastric acid secretion, they just reduce the protective barrier against the acid. So there are different ways that you can treat acid reflux. We talked about how excess acid can cause peptic ulcers, but they can also irritate the bottom of the esophagus. And so we want to treat that. You can treat it with direct antacids like aluminium hydroxide, and these directly neutralize the acid. You also have H2 receptor antagonists like ranitidine, and by inhibiting the H2 receptor, you're reducing gastric acid secretion. And then we have PPIs, which we already spoke about, and it's good to know an example, a name of the drug. So omeprazole is the one that I would remember. So even though gastric acid secretion, you'd think that the control of gastric acid secretion would be from the stomach mostly, but actually it's divided into a cephalic phase, a gastric component, and an intestinal component. So the cephalic component is what happens before this food actually reaches the stomach. So it can be triggered by the sight, smell, the thought, or the taste of food. And once the food's inside the stomach becomes distended, and you have lots of breakdown products of the food. And that's your gastric control of gastric secretion. And finally, you have some intestinal control as well, and that's triggered by the presence of breakdown products in the small intestine. So, yeah, important to remember that there are actually 3 things that control gastric acid secretion. And let's talk very briefly about some other secretions. So the pancreas, you'll remember, is both an exocrine and an endocrine gland. Its exocrine secretions are the enzymes listed here. So chymotrypsinogen and trypsinogen are both zymogens, and they're produced in an inactive form because if they were active within the pancreas, they would just break the pancreas down. So trypsinogen is released and it's activated by enterokinase. And that's a enzyme that's on the surface of the brush border of the small intestine. So trypsinogen can only be activated once it's entered the small intestine. And once you produce trypsin, trypsin then acts on chymotrypsinogen to produce chymotrypsin, which is a protease. The pancreas also produces amylase and lipase, and it produces iron, so sodium, potassium, chloride, and bicarbonate. And the bicarbonate in particular is really important because the kind that's just come from the very acidic stomach is going to be very, very acidic for the small intestine, which isn't designed for that. So the bicarbonate neutralizes the acid within the chyme. And some good clinical conditions to know about. So cystic fibrosis, you can get mucus plugs within the pancreatic duct, which block the pancreatic exocrine secretions from entering the small intestine. In chronic pancreatitis, damage to the pancreas itself can reduce the amount of exocrine secretions that it can produce. And if someone's had a pancreatectomy, for example, for pancreatic cancer, then they don't have a pancreas at all, so they can't produce the exocrine secretions. And for each of these conditions, you want to give the patient Creon. And Creon is basically a small pill that contains protease, lipase, and amylase, all in that pill. And it's just a supplement to improve, to kind of prevent malabsorption. Okay, and let's just talk about bile briefly. So the bile, as well as water, contains bile salts, cholesterol, and bilirubin. It's involved in the emulsion of fats, and it dissolves fat-soluble vitamins. And the fat-soluble vitamins are vitamins A, D, E, and K. Bile is produced in the liver. So this is a very simple diagram that I've just drawn here, and it's the kind of diagram that I would reproduce in an essay. So just talking through it very briefly, you have bile being produced in the liver, and it gets transported down the left hepatic duct and the right hepatic duct, which join to form the common hepatic duct. Then you have your gallbladder tucked underneath your liver, and the gallbladder is connected to the common hepatic duct via the cystic duct. And when the common hepatic duct and the cystic duct join together, they form the common bile duct, or CBD. You also have the pancreas here, and the pancreas has the pancreatic duct, which joins the CBD at the ampulla rafata in D2 of the duodenum. Okay, and something that I didn't understand until quite recently actually was how bile entered the gallbladder. Because if bile's draining down the common hepatic duct, down into the CBD and into the intestine, how does it flow back up into the cystic duct and into the gallbladder? Well, the answer to that is that at the ampulla of Vater, you have a sphincter of Oddi, and the sphincter of Oddi is usually contracted. So if all this bile is flowing down and it's not entering the intestine, it just builds back up, and the backflow is how the gallbladder fills up. And once it's full up, it concentrates and just stores the bile for when you need to release some bile. So how does the gallbladder and how does the pancreas know when to produce the exocrine secretions and when to release the bile into the duodenum? Well, cholecystokinin, or CCK, is produced from the duodenum and the jejunum, and those stimulate pancreatic enzyme secretion and gallbladder contraction. And the whole process of producing CCK is stimulated by protein and fat products in the duodenum. Let's talk a bit about absorption. So absorption takes place mostly at the small intestine microvilli. And it's the absorption of the breakdown products of digestion. So you've broken down proteins into amino acids and peptides, and those get absorbed into the microvilli. If you've absorbed peptides, you actually have intracellular peptidases that can cleave those peptides into amino acids. And when you break down fats, you produce fatty acids. Those fatty acids enter the microvilli and they get incorporated into chylomicrons. And these fatty acids, instead of entering the blood, they enter the lacteals. And the lacteals all eventually drain into the lymphatic system. So that's an important distinction to get your head round, is that most things are absorbed into the blood except for fatty acids. Then you have glucose and galactose, which are absorbed through secondary active transport via SGLT1. And the reason this is secondary active transport is that SGLT1 exchanges, it kind of uses the sodium gradient to transport glucose into the cell. But you need to actively transport sodium to generate that sodium concentration gradient, even if the transport of glucose or galactose itself isn't using energy. So that's why it's secondary active transport. And fructose enters the cell via facilitated diffusion through the GLUT transporter. And then you have water being absorbed at the small and large intestine. Okay, and the final stage is egestion. So defecation can be under voluntary and autonomic control. You have an internal anal sphincter and an external anal sphincter. The internal one's under involuntary control via the parasympathetic nervous system, and the external sphincter's under voluntary control via the pudendal nerve. And so when these stretch receptors in the rectum sense that the rectum's all full up and it's being distended, then that triggers the internal anal sphincter to open up, and that's involuntary. However, you don't defecate unless the external anal sphincter is also relaxed. So that's how you can kind of pick a good time to go, and you can choose when to relax the external anal sphincter. Okay, the final section of this lecture, I just have one slide on GI defense mechanisms. So because you're taking in food from the outside environment, there could be lots of bacteria, lots of toxins in the food, and so you need all these different defense mechanisms to protect you from those. So the first mechanism is taste. If you taste something bitter, then it's likely to be rotten, or if it's sour, it's gone off, so you don't want to eat it. So that's the first defense mechanism. Once foods entered the stomach, the gastric acid can kill bacteria. So that's another defense mechanism. And throughout the GI tract, you have an epithelial barrier where the epithelial cells are joined by tight junctions. And the tight junctions limit paracellular movement, and the epithelial barrier is a physical barrier itself as well. And then you have a mucus barrier, and we've already spoken about NSAIDs and their effect on the mucus barrier. So within the mucosa, you have a mucosal immune system. So you have IgA, and IgA is the immunoglobulin that's specifically found in the mucosa. And you also have lymphoid tissue. And I used to always get confused about the difference between MALT and GALT and Peyer's patches. But the difference is, so MALT is mucosa-associated lymphoid tissue. So it can be found anywhere where there's mucosa. So for example, in the nose, you have nose-associated lymphoid tissue, and that's NALT. In the lungs, you have bronchial or tracheal associated lymphoid tissue, and that's BALT. So MALT is kind of an umbrella term. And GALT is the MALT that's specifically associated with the gut. So gut-associated lymphoid tissue. And then Peyer's patches are aggregated lymphoid nodules, and they're basically a type of GALT. So Peyer's patches are a type of GALT, and GALT is a type of MALT. And it's important to bear in mind that these defense mechanisms don't kill all bacteria, because we have a microbiota in the gut, and that's full of commensal bacteria which are harmless, and we actually need them to aid digestion, and they also have a range of other functions as well. So the purpose of the GI defense mechanisms isn't to kill all bacteria, all pathogens. It's to ensure that the microbiota is commensals only. It's to get rid of the pathogenic bacteria. Okay, well, thank you very much. That's the end of this lecture. I hope you found it helpful. I'd really appreciate it if you filled out the feedback form, and these slides will also be in the feedback form if you wanted the diagrams or anything.